All right. Hello, everyone. Welcome to the 9th Annual MSU Science Festival. So happy to have you here. My name is Catherine Hagman. I'm with the Science Festival. I'll be your host for this session. And today we're joined by CIGLR. So excited to get started. So let's begin with our next talk. And we're talking all about harmful algae blooms. All right.
So hi, everyone. My name is Holly. I'm going to be talking about our monitoring program on the Great Lakes, monitoring harmful algal blooms. My goal of this presentation is to just give you guys an idea of what a day in the life of field work is for our scientists at CIGLR. And I'm really excited because field work is my favorite part of my job. I love going out on the boat, so I'm really excited to share that with you today. So to start out, harmful algal blooms are simply just an increased growth of naturally occurring phytoplankton. And this can be caused by environmental changes, such as in the spring when the surface waters warm, and when spring showers also not only bring spring flowers, but also they can bring harmful algae. So we start our monitoring program in May and June, and these pictures that I have here are images from our 2019 season when we had a very large bloom, and you can see the green surface of the water. You know, water usually looks blue, but here it's very, very pigmented green. And I have a little video here of the wake. You know, usually when you're boating, a wake is white, but this wake is green. That's how dense this algae bloom was in 2019. So when we're talking about harmful algal blooms on the Great Lakes, the main species that we're talking about is Cyanobacteria. We're talking about Microcystis, which is a single-celled cyanobacteria, so it's bacteria. And it's so small that you can't even see it with the naked eye. But when it blooms in very large colonies, large groups, you can see that green scum, and it changes the entire color of the water. Cyanobacteria is also known as blue-green algae. This bacteria actually has a pigment in it that fluoresces blue. So we have chlorophyll and the blue pigment, blue-green algae. And those pigments can actually be measured from remote sensing data. And these blooms can be so big that we monitor them with airplanes. We take an airplane and fly over the Great Lakes while we're also on the boat. And this is to help get a large picture scale of the size of the bloom and where it is. To give you an idea of size scale for this image, right down here, there's a big barge. So if you know how big those barges are, it might give you an idea of how big this green bloom is. So that's why we measure it with airplanes. The reason we care about this is because when one species of plankton blooms rapidly, it can change the entire ecosystem. Usually in the water, there's lots of different plankton that is food for zooplankton and fish. But when we have one species of plankton that's growing and taking over the entire ecosystem, it changes the food source for the larval fish, the bigger fish, and it can cause problems at an ecosystem level. In the Great Lakes, when we're looking at harmful algal blooms, we're also measuring a toxin that is produced by this bacteria called Microcystis. And that toxin is harmful to not only the ecosystem, but it's also harmful to humans too. We don't want to get that on our skin, or we definitely don't want to ingest it. And it can even get into the air and cause problems with breathing. And during a global pandemic, we definitely want to monitor these toxins in not only the water, but we're also hoping to add studies looking at the air quality too. And the biggest, or if you've heard of these blooms, you might have heard about the 2014 Toledo water incident, where that toxin actually got into the drinking water intake system and caused a lot of problems there. So our monitoring program helps keep an eye on where the bloom is, how big it is, and about that, how much toxin is in the water. So our lab is located in Ann Arbor, Michigan. And our lab primarily focuses on the monitoring the western basin of Lake Erie, as well as Saginaw Bay in Lake Huron. But through multiple collaborations with many different laboratories, harmful algal blooms are monitored binationally in all 5 of the Great Lakes. So during our HAB season, which I mentioned starts about in May, kicks up in June and July, we take out our research vessel in collaboration with NOAA, and we go out at bright and early, 6:30 in the morning, and we try to get out on the water by 8 o'clock. And so it's a long day. We're out there in the rain, in the hot sun, and it's a good time, but it's a lot of work. We have about 8 sites in Western Lake Erie, and then we also have 5 sites in Saginaw Bay that we try and monitor at all of those. So we visit each of those sites and collect water. So to collect water, we use a thing called a Niskin bottle, which is basically a big hollow tube with stoppers on the end, on both ends. And you cock those stoppers back, and then you can lower the Niskin bottle into the water, and you send a messenger down the rope to release a trigger that then closes the ends of that Niskin bottle, and that captures the water inside at whatever depth we're looking at. I'll pull that up, and then we collect the water in a big giant collection bottle. And one of these throws isn't enough water, so we have to throw it probably 4 or 5 times, and that takes about half an hour. So you can see here, cocking those ends back. And then we'll put it in the water, and there's a weight on the end of that. That weight is there to help keep the Niskin straight in the water column. And I'll lower it down to about 1 meter if we're looking at surface. So if we're looking at the surface water, we don't want to necessarily collect the absolute surface. We want to get a better generalized idea of the water column. So we put it down about 1 meter below the surface. You can see the messenger going down, and then it comes up and those stoppers have closed. And then the water is then collected. And you can see the coolers on the left-hand side. We keep the water in coolers after visiting all those 8 sites. And not only are we collecting water, but we're also taking a few other parameters at each site as well. So in the middle, you can see my coworker, Paul. He's throwing a fluoroprobe down through the water column, and that's looking at the entire column. So it starts at the surface and goes all the way to the bottom. And that fluoroprobe is measuring pigments. So I mentioned earlier that the cyanobacteria has a blue and green pigments. There's other colors, pigments in other different kinds of phytoplankton. So by measuring those different pigments throughout the entire water column, we can actually back calculate and figure out what kinds of phytoplankton are occupying different levels of that water column. It's pretty cool. And on the left, you can see my coworker Glenn using a hypergun. That's measuring surface radiance. That surface radiance is then used to help ground truth the data that we're collecting from the airplanes as they're flying over while we're collecting water. And that's important to take out atmospheric corrections. So when they're flying above us, there can be stuff in the air, and that hypergun can help give an idea of what's going on the water, so we can compare it to what's measured in the sky. On the right-hand side, you can see me filtering on the boat. I'm filtering for nutrients. Nutrients have to be filtered out right away to get out any life forms that are going to try and eat those nutrients or utilize those nutrients. So by doing that on the boat, we can take out the life forms so that those nutrients that are in the water at that time are then preserved at that time. After we're done sampling at all of our 8 sites, we'll take it back to the lab. There's an entire team of people waiting for us to bring that water back so that we can separate it out and measure for lots of different things, like different nutrients, such as phosphorus and nitrogen, or different pigments like chlorophyll and phycocyanin, the pigment in cyanobacteria. We're also measuring for DNA, like Nate was talking about. We're measuring for lots of different things. And that overall, even with an entire team of researchers, can take up to 3, maybe even 4 hours, depending on how much sediment is in the water, or how much growth, how much bacteria, cyanobacteria, or other phytoplankton is in the water. Can take a while. Here's a picture of us just filtering in the lab. We use many different sizes and take a lot of different samples. On the right, I wanted to point out that picture because Glenn is filtering in the dark. And the reason for that is he's measuring for chlorophyll, which is the pigment used in photosynthesis. So to stop that photosynthesis, we're taking that sample in the dark, so photosynthesis isn't going to happen. He's using a green light because the phytoplankton are actually kind of colorblind to that green color. So he works in the dark, but uses that green light to be able to see. So overall, this data is compiled and used for many different things. It's all publicly available data. But one of the products that we put out is our Lake Erie HAB tracker. It's available on the CIGLR and CIGLR website. And that's a time series animated, almost real-time visualization of showing where the bloom is and how big it is. And that can be used by fishermen or other boaters or even other stakeholders to figure out where the bloom is. We also post the data about the toxins that we measure on there, so we can keep the public informed about these blooms and help protect our ecosystem and protect even just going out for a day on the boat and having a good time, so they can avoid that bloom. And with that, I'll open it up for questions.
Great, Holly, thanks so much. That was really fascinating. Thinking about the effects of these blooms, I had never thought about air quality. So fascinating. Thanks again to everyone who's joining us. Just as a reminder, if you're joining us from the Zoom webinar, feel free to use the Q&A function at the bottom of your screen to ask any questions. I'll be sure to read those off for Holly. And if you're joining us on the Facebook Live, feel free to enter your your questions into the comments section, and I'll read those off as well. And Holly, I'll start off with a question I have for some of our audience members who maybe joined for your talk but missed the first talk of the day. Could you tell us a little bit about CIGLR and what it is that you do?
Yeah, so CIGLR is the Cooperative Institute for Great Lakes Research. We actually partner with the NOAA facility, which is the National Oceanic and Atmospheric Administration. And our Cooperative Institute partners with them to monitor and study everything about the Great Lakes, from water quality to harmful algal blooms to DNA and fish. We do it all.
Great, and we have one question. Someone wants to know, what happens if I drink water with algae?
It depends. So if you drink water with algae, it's not necessarily bad unless that algae is in high concentration and if it has that toxin in it, it can be harmful to your health. Microcystin is actually a liver toxin, so it'll affect your liver. And you'll see that pretty much right away with gastrointestinal issues. Can have if in higher concentrations, that toxin, if ingested, can lead to liver failure, and it has been known to cause death in dogs. And I don't recommend drinking it.
Yeah, I guess not, huh? Great. Looks like we have time for just a couple more questions. So if you have a question for Holly, again, feel free to enter it into the Q&A function at the bottom of your screen on Zoom or in the comments section on Facebook. While we wait to see if we have any more questions from the audience, Holly, I'm just curious about what got you into this line of work. What was your background? Like, what did you study in college?
That's a great question. I studied everything under the sun in undergrad. I did my undergrad at the University of Alaska Southeast. So I got a lot of experience working with whales, working with other marine mammals, and I did a study on marine debris. And I also worked with mariculture and the growth of farmed seaweed. And I realized through all those different things that I really just wanted to study plants. I really didn't want to study animals. So in grad school, I studied a toxin called domoic acid in Africa. We were studying documenting the first, making the first documentation of this a different type of harmful algal bloom in a remote area in Africa. And so it's the same type of measuring that toxin is the same in many different ways. So I was able to transcribe my skills from measuring one toxin to another toxin to microcystin in the Great Lakes. So I'm fully marine background. This is my first freshwater gig. And it's great because when I go out on the boat and get all wet, I don't have salt stains on my clothes.
Oh, that's great. Gotta love our Great Lakes. So one last question for you before we take a brief break for our next presentation. You mentioned that your studies are based on Lake Erie. Are there other bodies of water that these algal blooms can affect?
Yes. So these harmful algal blooms are also, they occur in small lakes too. And we as CIGLR mostly focus on the Great Lakes, but there are other like USGS and other entities that are studying inland waters and the effects of harmful algal blooms in those small inland waters.
Good to know. Great. Well, thank you so much again, Holly, for joining us. And thank you everyone for joining us as well, tuning in. We're going to take a brief break and then we'll be back right at 1:30. I'm going to be talking about Asian carp. So stay tuned and thanks again.